Various techniques are involved in the cell biology. Very first technique is microscopy. From name it is clear that microscope is used, which is an instrument which was invented almost in 1609 by Galileo Galilei, who was an Italian astronomer in Pisa, Italy, and he has been called father of observational astronomy. And another person who was an optician, and he made optical instruments and spectacles as well. He was Zacharias Janssen. So both these, Galileo and Zacharias Janssen, contributed a lot in the field of microscopy because they all the time were busy in lenses. So in microscopy, lenses are used in order to give a magnified image. A magnified image here means that a bigger or a larger image, which helps us to easily observe the cell. Dear students, in microscopy, two aspects are involved. One aspect is magnification, and the second aspect is resolution. Magnification means how bigger an image could be, and it is represented by symbol X. For example, if an image is 10 times bigger than its actual size, then we write it as 10x. If it is 100 times bigger than its actual size, we write it down as 100x, or you can call it x10 and x100 as well. So magnification can be defined as the factor by which image is to be enlarged. This means that an image can be enlarged up to our desire, that how much bigger image we want to clearly understand the inside of the cells and to know about the nature of the cells. Basically, two convex lenses are involved in microscopy. One convex lens is in the eyepiece area, that is called as eyepiece lens, and the second lens is the objective lens in the microscope. In order to understand magnification, I am giving you an example that if it is written as objective lens power is x10 and eyepiece lens power is x10. So what is the magnification of that image? That how much bigger or larger image is made? We simply multiply these two objective lenses powers, that is x10 multiplied with x10, and magnification of that image will be x100. Likewise, if our objective lens power is x10 and our eyepiece lens power is x6, so by simply multiplying it, we can get the result, and that result is 240. This tells us about the magnification power of the microscope. The second aspect, students, which I have to discuss with you is the resolution power of the microscope. As I have told earlier, that microscopy involves two basic terms or things or phenomena. One is magnification, that how bigger an object can be made, and second is resolution power of the microscope. This is also known as resolving power of the microscope. Another aspect is resolving power. So how can we define resolving power? This is the ability to distinguish close objects as being separated from each other. So resolving power is also called as resolution power. Mind it, this is the limitation of a microscope. I can explain it to you in a very simple way, that if you make these two circles, or you can assume them as dots, these are separated from each other, and a clear visible difference is that you can differentiate between these two balls. But over here, distance is reduced. In the next step, more reduced, and it is more reduced distance that you can hardly separate between, or you can hardly distinguish between these two balls. And in the very last image, you can see clearly that it is a blurred structure or a blurred image. Likewise, we can explain it from clear to blurred image. So this is the limitation of a microscope. Magnification is good. You have magnified the object well. Second aspect is the resolving power of the microscope. So resolving power can be the limitation of a microscope, because at certain extent that microscope won't be differentiate between two very fine or two close objects. So that's why we define it as, this is the ability of a microscope to distinguish close objects as being separated from each other. When blur image appears, this is the limitation. Likewise, human eye differentiates between 2 points at least 1 mm apart. This is the limitation of human eye. Similarly, if we compare it with the light microscope, it gives us image 500 times more bigger than the human bare eye. It means naked eye of human being can differentiate between two close points which are maximally, or you can say minimally, which are present at this distance, that is 1 mm. But if we compare it with the light microscope, light microscope's resolution power is 500 times more than the human naked eye, bare eyes limitation, or the resolution power. So we can better get an idea by comparing powers of various microscopes. For example, the very first microscope is TEM, that is transmission electron microscope. It gives us 2D images, and its magnification power is 1 million times than the size of the real object, the real size of the object. Fine. And the second one is resolution, that is 0.14 nanometer. Second is SEM. SEM is that microscope which gives us 3D images. That is called a scanning electron microscope. Its magnification power is 100,000 times than the real size, and its resolution is 0.2 nanometer. Dear students, another microscope is called as stereo microscope. In layman terms, the stereo microscope is also known as dissecting microscope, and its magnification power is 7.5 to 75 times more than the real image. It can magnify the object 7.5 to 75 times more than the real size. And its resolution power is 100 to 120 nanometer. And the last one is simple microscope. Simple microscope's maximum resolution power is 1500 times. Its magnification power is 1500 and its resolution power is 200 nanometer. So this is a very simple comparison. Like, this is the simple one. Comparatively, this is more better. This is normally called as dissecting microscope because it is used in labs for dissection. Then there is SEM, and highly advanced is TEM, most powerful microscope.